My name is Isabel Davis. It is week 3 lab exercise of bacterial morphology and staining techniques. So I have all my stuff set up here. I have my slides labeled, and I'm going to start with some broth used in a previous experiment. I did forget to light my candles. Let's get my candle lit because we are using sterile technique. Okay, so I have my candle lit. I've got my these here for my bacteria in my broth, and this is what I'm going to use. And I'm gonna clean it off. Here it is right here in alcohol, sterile, and I'm gonna clean it off, make sure it's dry prior to entering it in here. So I'm going to sterilize prior, and I'm starting with the S. cerevisiae. Again, it was used in a previous lab. All right, now I'm going to get some of the bacteria in here and proceed to place it on my slide. Okay, and then I'm going to toss that. I'm going to re-sterilize, and I'm going to cap it. And I'm going to continue this process with each broth. This is E. coli again, used in a previous lab. Going to get my second one. Sterilize. Make sure it's completely dry. Get some of this bacteria in here. Place it on the E. coli slide. Then we're going to use we're going to re-sterilize. Close that up. Then we're going to continue with the S. epidermidis broth. Collect some of that and place that on this slide. Toss that, re-sterilize, and close that up. Okay, so I'm gonna let that dry for about 10 minutes, and after that, then I'm going to do something called the heat fix it technique, which helps the bacteria stick to the slide, to the slide in the gram stain. And then after that, I'll let it cool for 10 minutes. But I do need these to dry for at least a few minutes. And I don't want to do it too close to the flame, because then it'll burn it. Like I used to do this in the laboratory. I worked as a lab assistant, and we did gram stains all the time. But I never really fully understood why we did it. What was the purpose of it? I don't know, honestly. I wasn't too sure why we did the gram staining. I just know I did it. I let it dry. And then the clinical lab scientists, they read the stain. We stained, we would look at white blood cells on stool samples and all kinds of good stuff. So pause. Okay, so then what we're going to do is oh, here, I guess I can use these, huh? All right. And now I'm going to continue with my gram stain that are done usually in laboratory settings. I'm going to be using Crystal Violet. This is the one I'm going to start with, is Crystal Violet, and I'm going to place 2 drops on here, and I'm going to wait for 60 seconds. Okay, that's 2 drops. And these I'm going to just do all of them because it's actually faster. Well, this is how I would do it in the lab. Do all of them, and then I'd gram stain all of them. Rinse them off after a minute. So I'm gonna wait that minute, and then I'm gonna rinse them off with tap water. And so what I'm going to do is I'm going to So this is an easier method, so the water is able to just rinse off after the minute. And then I'll continue to do it with the other sides, so I can rinse them all at the same time. I learned this with the lab. And the Crystal Violet, the Crystal Violet stain is associated with peptoglycan. And what that is the bacteria has peptoglycan in the cell, and that is why it stains purple. Okay, so it's been about a minute. We're gonna rinse that off. Okay, rinse that off there, rinse that off. Okay, and then after that, and then I'm going to continue with the iodine, 2 drops, 2 drops. Okay, that is what I'm going to do with iodine, and I'm going to wait 60 seconds for the iodine to dry. And with the iodine, so the way the iodine works is the iodine it has a negative charge, and together with the Crystal Violet, because the Crystal Violet is a positive charge, they form a complex called a Morton, and it helps the peptoglycan and the Crystal Violet to link to the peptoglycan in the gram-positive cell walls, and they have a thicker cell layer, so that's why we use the iodine. It makes it stick. And that'll stay for 1 minute. And then we'll go rinse these off. Okay. Rinse that off. Okay, and now I'm going to proceed with the safranin, and this is 2 to 3 drops of this. And we're gonna let that sit for 30 seconds. This stain is called a counter stain because it turns it pink in color, helps distinguish between the 2 types of cells. After the Crystal Violet, it'll stay in the granite cell wall. The ground cell walls take in the saffron, and this makes their cells walls pink. So that's how it gets the pink color. So we're gonna wait 30 seconds here. And then we're gonna proceed to rinse that off. Okay. So then we're on the last ones. And this is going to be the decolorizer, which is the one we're going to use last. Okay. Let that sit. I'm going to rinse those off. And then now what I'm going to do is I'm going to get my samples. I'm going to let them dry completely, and then I'm going to look at them underneath the microscope and upload photos. Thank you guys.